Hello, APBIO. In this last bit of notes, I'm going to talk about a little bit more about prokaryotic gene control. So today in class, we talked about eukaryotic gene control and the importance of eukaryotic gene control that allows cells not only to respond to the environment, but allows for cell specialization. So that's important in eukaryotes. In prokaryotes, we explored the operon as a mechanism of control in prokaryotic cells. So we looked at that in our lab. That was the PGLO lab. And we looked at the ERA operon, which is an inducible operon. We also chatted about the lac operon, which is an inducible operon. But many operons are also repressible operons. So they're in the on position, essentially, as naked DNA. And then they can be turned off in the presence of something. And that's what the trip operon is. So it's called the trip operon, the TRP operon. So I'm here in your notes. And the purpose of the trip operon is to code for enzymes needed for the synthesis of tryptophan. So if you can recall from our conversation in biochemistry, and then again looking at protein synthesis, tryptophan is one of the 21 amino acids. So organisms need it to be able to make proteins, and it's not an essential amino acid for bacteria. They can actually make it, but if it's in the environment, it doesn't make any sense to make it, right? So this question number 11 asks, what is tryptophan? Why do organisms need it? You could answer that question. And then it's tryptophan and the trip operon is a repressible operon. So it's opposite of an inducible operon. So let's think about what happens. The repressible operons here, so let's orient ourselves. Here we have the promoter. Remember, that's the operator. Here's the operon right here. Here's the promoter. That's where the RNA polymerase is going to bind. And then here's the repressor protein, this pink thing right here, that's bound to the operator. And then here's all the structural genes. So if you notice, in the presence of tryptophan, so in this case, these are repressors. This specific small molecule, in this case, because it's the trip operon, it's tryptophan. So this amino acid binds to the repressor protein, and it activates the repressor protein while it's bound. So before, with the lac operon and the arabinose operon, remember, in the presence of arabinose, this pulled off or allowed for the binding of RNA polymerase in the presence of arabinose. But in the repressible operons, the presence of a molecule is going to inhibit the binding of an RNA polymerase. And you can see this guy here is trying to get on and bind to the promoter, but it can't because the trip repressor is active. The repressor protein is active. Now, when tryptophan is low, this trip repressor protein, the trip repressor, is going to be inactivated, meaning it's going to pull the repressor protein off of the operator, and now our RNA polymerase can bind and transcribe the genes. So it acts the opposite. So if we think about how the regulatory gene is synthesizing this repressor protein, in repressible operons, the repressor protein is synthesized in the inactive form. It's not active. The only way it's activated is in the presence of something else. So this molecule here activates and inactivates this protein, right? That's the opposite from inducible operons. In inducible operons, let's go back and just look quickly at the lac operon. In the lac operon, it's inactivated in the presence of something. So it's inactivated in the presence of something, and here it's activated in the presence of something. So let's go back and look again. This is inactivated in the presence of something. So it's pulling it off in the presence of something. And this is making it stay on in the presence of something. So the mechanism is opposite. So in repressible operons, the synthesis of the repressor protein is in the inactive form. But in inducible operon, the synthesis of the repressor protein is in the active form. You see, this is an active repressor, and it binds directly when in the absence of something, but it is induced when the specific small molecule is in the environment. So in the repressor protein in the presence of tryptophan is going to be activated, because why would you make tryptophan if you already have it? That's silliness, right? And in the absence, the repressor protein in the absence of tryptophan is activated. It comes off of the DNA, and it is, it's inactivated. It comes off of the DNA, and now RNA polymerase can transcribe those genes, bind to the promoter, transcribe the genes, and then make tryptophan so that those bacteria cells can then make proteins that have tryptophan in them. Okay, that's it. That's all for your notes for this exam, and I hope that was helpful.